Welcome everyone assistants. My name is Macarena and I will give you the introduction to Nataila Pigni from University of Connecticut, who will give us the talk entitled Light on Carotenoid-protein Dynamics in Photosynthetic Organisms, the case of OCP. Now I will give the instruction of the talk. Please keep the microphone off during the entire presentation. A question can be asked at the end of presentation. By Zoom, you can raise your hand or write in the Zoom chat. And by YouTube, you can write them in the chat too. They will be notified when they can activate my microphone. Now, I leave you with the Dr. Álvaro Muñoz, who will perform the official presentation of the talk. Thank you for attending this cycle of talk.
Thanks, Macarena, for the nice introduction. For me, it's a pleasure to introduce to Nataila Pigni. She is from Argentina. She started her career in science, doing a licenciatura en biología molecular at the University of. Then she moved to 18 months to work at the Universidad Nacional de Cuyo at Mendoza to continue to carry on some studies in genetic and morphological traits of wild potatoes. And then she moved to Barcelona in 2008 to get her PhD in drug research, development and control, working on natural products chemistry in Jaume Batista Group at the University of Barcelona. In Europe, she did a short-term research stay for 3 months in plant metabolics at Max Planck Institute of Molecular Plant Physiology in Germany. In 2014, she moved back to Argentina to get a permanent position as an assistant researcher at CONICET and as assistant professor at Universidad Nacional de Córdoba to work in food chemistry in order to address some research in polyphenolic profiles of different seeds. Always restless, she became interested on the study of proteins, polyphenols, interaction via in silico method as an approach to explore their bioactivity. During 2018, she got the knowledge to the subject by spending 4 months as a visiting scholar in Javier Luque group at the University of Barcelona, Spain. Since 2019, she became a very happy mother and then worked in the José Gascón group at the University of Connecticut and the United States, studying the photophysics of carotenoids related to proteins and to get insight into the different processes related to the energy capture of plants and different algae. So this is the topic that she will talk today. In the near future, she plans to return to Argentina by the end of this year to incorporate computational chemistry method to the food science at one research group at CONICET, and also to try to improve the political determination in terms of the further of national decision in food quality and different aspects. So, Natalia, the audience is all yours. Thank you.
Thank you. I will share screen. Yep. Just a moment. Okay, can you see it properly? Well, thank you for the nice presentation, and especially thank you for inviting me to participate in this webinar. It's really important to be connected between us. So a big thanks to the Red Latinoamericana de Físico-Química Teórica for leading this wonderful initiative. Well, as you said before, I am going to present our work focusing on the orange carotenoid protein. It was performed in Gascon lab in US. First of all, just a brief mention to where I belong. As you listen from my presenter, I have a very diverse background. I've worked in plant molecular biology, natural product chemistry, food chemistry, and more recently in of Cordoba, which is in the center of the country. And once there, at some point, I became interested in approaching the study of bioactivity through computational methods. So I started my journey to computational chemistry. In 2019, I had the great opportunity to start collaborating and working as a postdoc in Gascon lab. And I am going to talk today about one of the main projects which have been working here. So the protein which I'm going to talk today is related with the defense mechanism that from the cyanobacteria. So as you probably know, photosynthetic organisms such as plants and cyanobacteria take advantage of the solar energy to convert inorganic matter into organic compounds. Under normal or low light conditions, as you can see here in the picture, they perform an efficient energy capture. However, under high irradiance conditions, there are some damaging species that are generated, such as the reactive oxygen species, and this represents a potential damage to the cells. So these organisms have developed different defense mechanisms. In particular, the protein that we are going to talk today, which is OCP, participates in a mechanism which is known as non-photochemical quenching, and basically it means that these proteins that are involved in this excess energy dissipation in different forms, such as heat. Yeah, sorry. Yeah. Well, the orange carotenoid protein is a water-soluble protein. It has a size of around 35 kilodaltons. And as you can see here in this representation, it has two well-defined domains. One is the NTD, which you can see in orange, is an all-alpha helical, it has an all-alpha helical secondary structure, and it's known as the effector domain. The other that you can see in cyan, represented in cyan, it's the CTD, the C-terminal domain. It has the function of a sensor domain, and it's a mixture of alpha helices and beta sheets. Encapsulated between these two domains, we can see the carotenoid, a carotenoid molecule, and OCP in general can bind different types of carotenoids, but there is one requirement for the protein to be photoactive, and it is a carbonyl group on the beta-1 side, on the CTD side. This carbonyl group is key because it interacts through two key interactions, which are hydrogen bond interactions with a tryptophan and a tyrosine residue on the CTD. These residues are highly conserved. And on the other side, on the beta-2 side, or the NTD side, it interacts through hydrophobic interactions, also with tyrosine and tryptophan residues. OCP in its inactive form is characterized by an orange coloration, and after the excitation by blue-green light, it becomes in its active form, which is known as OCP-R. The R is because of the characteristic red coloration of this form of the protein. And as you can see here on the plot, both forms of the protein have essentially different absorbance spectra. So this is key, but because it's like a characteristic of each of these states. It's key to study the system. Upon photoactivation, there are three main events that occur. The first one is the breakage of these H-bond interactions that are on the beta-1 sites. And this first step allows to the second event to occur, which is a 12 angstroms translocation of the carotenoid into the NTD site. And once the carotenoid is translocated, it occurs, some structural changes occur that lead to the domain separation. So that compact state that we saw in the previous slide, it's like finds an open position to exert its biological function. Still today, there is no model of the red form of the protein available. However, in 2015, Leverens et al. Reported the crystal structure of RCP, which is the red carotenoid protein, and is basically the isolated NTD from the activated OCP. So it has the carotenoid already inside the NTD site. It has moved to the NTD site. And the most interesting feature of this protein is that its UV visible spectrum is almost identical to OCP-R. If we superimpose both structures, the structure of OCP and the structure of RCP, we can see that the helical structure of the NTD, it's almost the same. It has, of course, some changes occur to allow the carotenoid translocation, but they are essentially the same. More recently, in 2019, Connell et al. Published this interesting work in which they used time-resolved spectroscopy, combining different techniques to reveal the timing of the photocycle of this protein. So basically, the proposed mechanism implies that after a photoexcitation of the orange form of the OCP, it occurs a distortion of the carotenoid that leads to the breakage of those hydrogen bonds on the CTD site. This part of the mechanism is the fastest part of the mechanism. It occurs in picoseconds, and it leads to the formation of what is known as P1, photoproduct 1. Just a moment. Well, this photoproduct 1 has the characteristic to have the H-bond, these key H-bonds broken, and it's also, the carotenoid finds a more planar structure, which leads to a better conjugation of the system, and it explains the red shifted spectra, absorption spectra of this photoproduct. After around 50 nanoseconds, a percentage of this photoproduct 1 follows on towards the continuation of the mechanism, and it produces a small displacement of the carotenoid to lead to the formation of photoproduct P2. It is important to say that about 60% of P1 reforms the H-bonds and return to the dark state, which is the orange state. After around 500 nanoseconds, or I mean, in a time scale of 500 nanoseconds to 1 microsecond, the NTD side, the protein, undergoes some structural changes that are still not known, and it results in the formation of P2', another intermediate photoproduct, that then, after 10 microseconds, follows on to the formation of P3, photoproduct P3, which is the compact state, but the carotenoid has been fully translocated into the NTD. The most important feature of this photoproduct P3 is that it has already similar spectral features of the OCPR, the active state. So it means that the protein environment that the carotenoid has in this state, in P3, is basically the same that it has in OCPR, in the active form of OCP. The active form of OCP implies the opening of those domains, as we can see in this picture, and it happens in a much longer time scale of more than 1 millisecond. Though all these intermediate photoproducts, P1, P2, and P3, have already redshifted spectra with respect to OCP0. And as the word indicates, it's a cycle. So if we leave OCPR in darkness, it spontaneously will come back to OCP0. But also in vivo, there is a protein that helps and accelerate this process, which is known as the fluorescence recovery protein. So in summary, despite the progress in describing many aspects of this mechanism, there are still a lot of, many open questions about different parts of the cycle. One of them is, which are the changes that the absorbance of light triggers, which are the changes in the carotenoid that leads to the breakage of these key H bonds, which is the structural dynamics of the carotenoid translocation, and also the structural dynamics of the domain separations. With all these questions in mind, we starting approaching the event of the process of carotenoid translocation. So now I will show you which was our approach to tackle this question through computational methods. So first of all, our approach, the first part of our approach was to validate the reference models that we have available. The reference model were the OCPO state and the RCP, which is similar to the active OCP. And we have the crystal structures of these proteins are available, and they are completely characterized through UV visible spectroscopy. So as you know, the crystal structure represents only one possible configuration of the system. So the system in solution or in vivo, in reality, exists in an equilibrium between multiple conformations. So to explore and to get a better sampling of the different conformations, we performed molecular dynamics of both systems, of course, solvated and properly prepared, using the Schrödinger platform and the tool to run molecular dynamics, which is Desmond. And having these values of reflecting the excitation energies of these two reference models, we decided to perform QMMM calculations in order to see if we could get those experimental values reproduced in our calculations. So we extracted snapshots from the molecular dynamics that we ran, of course, after doing the proper analysis of those molecular dynamics. We extracted snapshots evenly distributed along the molecular dynamics simulation, And we performed a QMM local optimization of the carotenoid, keeping the protein frozen. All of this was performed using the QSite, which is implemented also in Schrödinger platform. And after this local optimization, which of course considers the protein environment of each of the configurations of the molecular dynamics, we calculated the excited state energies. Again, using the QSite tool from Schrödinger and using considering the protein solvation effects using the Poisson-Boltzmann implicit solvent method, and using CAMB3-LYP. We did this calculation at the CAMB3-LYP level because it is known to be to reproduce correctly the excitation energies. The first question that arises from this approach is why to perform a local optimization? Why not to use directly the snapshots that we extracted from the MD? And basically, the answer to this is that the force field used in the molecular dynamics in general, we know, it is known in the literature that the force fields overestimates what is known as BLA, bond length alternation. Excitation energies of this type of compounds, like the carotenoid that we see here, are related to the conjugation and planarity of the system. And one of the parameters to measure the conjugation of the system is bond length alternation, BLA. And the BLA, it's directly, I'm strongly correlated with the excitation energies of the system, and of course with the lambda maximum values, as we can see here in the plot. So we need to perform this optimization to have the correct BLA and a correct estimation of the BLA and the excitation energy. This optimization generates just only minimum displacement of the polyene chain, and as it is done with the protein environment frozen around, it's basically the same configuration, and we just adjust the BLA. Our results are presented here, are summarized here in this slide. Here you can see the circles, these circles that represent the calculated excitation energies or lambda maximum values for each snapshot along the molecular dynamics simulation. This is for OCPO and this is for RCP. And we calculated through one sample t-test the 90% confidence interval of these calculated values. And you see this confidence interval represented here as a band, an orange band for the OCP values and a red band for the RCP values. And as you can see here, the experimental measured values of these absorbance falls exactly or almost in the middle of these confidence intervals that we calculated with our values, with our approach. So once we had these models, these reference models validated, we continued with the main goal, which was to explore the carotenoid translocation process. The OCPO state, in its orange state, is a thermodynamically stable complex, and the carotenoid to be translocated needs to overcome certain energy barriers. In nature, this energy is given by the photon absorption, but in this case, where we wanted to bypass the creation of photoproduct 1, and we only wanted to model how the translocation of the carotenoid occurs through the protein cavity, we decided to use, we had to use enhanced sampling methodologies. Having in mind the model of RCP as the final state of the process, of the translocation process, then we started with OCPO and we performed, we developed a well-tempered metadynamics procedure to drive the carotenoid translocation. Basically, what we wanted is to pull an invisible thread to move CAN towards the NTD. So for this, we had to define a collective variable, a distance to play around. So a coordinate for this translocation to occur. And in our case, after trying different variations, we found that a good collective variable that defines this is the distance between 5 residues on the outside part of the NTD and 1 carbon on this side of the carotenoid. So we just attached a thread here and we try to pull the system, the carotenoid, through into this direction. As we had the QMMM methodology already validated, we now can combine the metadynamics procedure and with the QMM calculations to characterize the spectral signatures of the different states of the translocation process. So we performed the metadynamics and we extracted snapshots evenly distributed along the time of this metadynamics. And from there, we performed the same approach of QMMM calculations to obtain the excited state energies during the translocation of the carotenoid. Here you can see the movie of the carotenoid translocation, and on the right you will see how the absorbance maximum evolves during the translocation of the carotenoid. As you can see here, during the first 48 nanoseconds, the protein remains like in its orange state, and then it moves, it jumps to the red state. We will see here in the next slide like a more detailed view of this process. Here you can see the dots here in this plot means the excited, the lambda maximum values calculated for each snapshot of the metadynamics. Then we have the average, the moving average calculated here shown here as the line, and the bands, the orange band and the red band, which are the calculated lambda maximum values for OCPO and the red carotenoid protein. And on the right, you can see the different states, representative structures of the different states of this metadynamics. This is an upper view of the protein, in which the protein is represented as surface, so we can see the protein, the cavity of the ligand and the solvent accessibility and the solvent accessible areas here. And this snapshot represents the initial state, which is OCPO. Here we can see that the carotenoid is still H-bonded to these two key residues on the CTD side. And the structure of the carotenoid here is clearly seen as bent, a little bent. So we don't have the planarity that we need for a redshifted state. An important thing to remark here is that all of these times that you see here are metadynamics times. So this time do not represent the real kinetics of the process. It is only used as a reference of the different states of the system throughout the pathway for the translocation. After the 48, 49 nanoseconds, there is a movement of the carotenoid towards the NTD. And this snapshot represents that movement, and clearly it can be seen that the carotenoid is more planar, which explains it favors a better conjugation of the system, and it explains the redshifted state, and it has been moved halfway through the towards the NTD side. After this, we see a decrease of the absorbance, in which this is what we call like the bottleneck of translocation. And what we infer from this is that at this point, the carotenoid, the protein cavity makes more difficult for the carotenoid to accommodate a planar configuration. So we can see like big distortions here in this snapshot of the big distortions of the carotenoid that lead to blue shifted conformation. And finally, by the end of the metadynamics, we reach the point of the 12th Armstrong translocation. The carotenoid is almost completely immersed into the NTD site. And we can see that this cavity of the protein can accommodate a much more planar configuration, and it explains the redshifted state. If we remember the timeline of the photoactivation, during the photoactivation process, we had two intermediates, which are P2 and P3. P2 is an intermediate state of CAN translocation, is already redshifted, and P3 is the full 12th Armstrong translocation previous to the activated state. Here, with these models that we see here, and analyzing the literature and the experimental evidence, we can say that these models are in agreement with the proposed photoproducts P2 and P3. This intermediate photoproduct, P3, as you can see here, the carotenoid has been completely translocated into the NTD. It reached the 12th Armstrong translocation that we were looking for. And it's the previous, it's important because it's the previous state to domains, for domain separation. So it's a really interesting model to approach the following step of the mechanism. And here on the right, you can see a picture of OCP P3 overlapped with the red carotenoid protein. And we can see that both models share the same protein, the same ligand cavity here. And the only differences are in the extremes of the carotenoid, where the carotenoid can rotate. It's not weird to see this, because in RCP, both sides of the carotenoid are more exposed to solvents, so there they can an enhanced freedom to rotate and to find different conformations of the system. Well, the highlights of this work were that we achieved an accurate reproduction of the experimental spectral features of the reference models through this combined methodology that I've shown, combining molecular dynamics and this QMMM approach. We proposed molecular models for these two intermediate photoproducts, P2 and P3, and we got this OCP P3 model to propose this as a good starting point to explore the subsequent steps in the activation mechanism. So now I would like to, before closing the talk, I would like to show you just a brief outlook of our results on the next chapter of the mechanism, which is starting from OCP-P3, exploring domain separation, the domain separation process. This work has been done in collaboration with Dr. Sharawi and Dr. May from the Molecular and Cell Biology Department here in UConn. And and it's almost ready for submission. It's going to be uploaded within these days to the Chem Archive. And just a brief outlook of what we did. First of all, just as a reminder, OCPR in its active state has its two domains opened. And they are open because they need to be open to interact with the phycobilisomes in the cyanobacteria in order to quench the excess energy, to exert its biological function. As you can see here in this model of the binding of OCPR to the phycobilisomes, we can see that this is in red, it is represented the NTD site with the carotenoid, which is the one that is going to interact with the phycobilisomes, and the phycobiliums, which are the pigments that are in the phycobilisomes. And attached to this domain, we have attached through its flexible loop, we have the C-terminal domain here. So it needs to be open in order to interact with the phycobilisomes. So in this work, in our work, we approached the domain separation again, taking advantage of a metadynamics methodology, and starting from this intermediate photoproduct in which the carotenoid has been already completely translocated into the NTD, we defined, in this case, two collective variables for the system, two collective variables to try to explore domain separations. One of them is the distance between this alpha helix on the NTD, on the N-terminal extension, and the CTD domain. These interactions need to be dissociated in order to allow domain separation. And the second collective variable was the distance between the center of masses of these the two domains of the protein. And what we found as the one of the most interesting results is that when we compare the same procedure applied to OCPO, we found that the domain separation is only favorable on once the carotenoid has been translocated. The domain separation is not favorable when the carotenoid has not been translocated, as in OCPO. And here you can see the free energy surface of the domain separation starting from P3. P3 is here represented by the A as an associated state. And you can clearly see that the global minimum for OCP-P3 is the dissociated state, which is state D, which we propose that is a model of the active OCP. So we have calculated the minimum free energy path, and we can say that the most the most probable path to domain separation starting once the carotenoid has been translocated is the opening of this and the dissociation of this alpha A, which is here in red, and then the domain separation towards the dissociated state. And regarding this dissociated state, this hypothetical model of OCPR, we have we have evaluated this model through equilibrium molecular dynamics, and we can say that it is a dynamic structure that fluctuates through different orientations of the domains, and it has a solvent accessible area increased, which is expected if the domains are separated. We can see the water molecules here in the middle between the two domains. The contacts with the loop and the NTE, these N-terminal helixes, are kept. Even this is a separated state, this flexible loop is not completely extended as someone can expect, but it remains in contact with the two domains. And this, the most interesting part is that this is in agreement with experiments, of course. And what we think is that probably this dynamic structure in vivo, the biologically relevant conformations are selected by the interactions with the other proteins, with the proteins that are present in vivo in the cell. So the still unknown, the still open question regards to the primary photoactivation event. We still not know what happens here in these earliest steps of the photoactivation mechanism. What triggers what is the earliest thing that the light triggers? And there has been some hypotheses, such as the isomerization at different parts of the carotenoid, of the polyene chain of the carotenoid. And a very recent publication of this year have proposed a proton transfer event in which one of the two residues, or the tyrosine or the tryptophan, transfer a proton to the carotenoid, and it leads to the formation of an unstable oxocarbenium ion that is at some point is distorted, generated the following changes in the mechanism. However, well, we are working on this now, and we have an interesting road work ahead. Well, that has been my talk. I would like to say thank you to the audience for listening this. Of course, a big thanks to my group, to Jose Gascon for the opportunity to work here. Also to my colleagues, Kevin and Kidmini, they are PhD students in our group. I would like to say thanks to Dr. May and Dr. Sharawi for the collaboration on the domain separation work, and to the National Science Foundation for the funding, and of course, to CONICET and my institution in Córdoba for allowing me the possibility to spend a long time here learning about this. Thank you all.
Many thanks, Natalia, for your nice presentation. So it's a pleasure to have you here, and thank you for your contribution to this network. So now it's time for questions. So if anybody have one question, can raise the virtual hand and open their microphone. We have one question from the YouTube audience. That question is the following. Hello, what parameters do you use for the Gaussian potential applied in metadynamics, and how could you validate the MTD trajectory?
Yeah, let me see if I have a snapshot. Yeah, well, let me go back. So I will stop sharing this and I will share the screen so I can show you the parameters of the metadynamics. Share screen again. Yep. Yeah, I have this open. Yeah. The parameters that we used are here. I did not add them to the presentation just because to not do it longer. But the width of the Gaussian potential was 0.06 and the initial height 0.03 kilocalories per mole. The deposition time was 0.2 picoseconds, and the bias factor, because we used well-tempered metadynamics, was set to 10. We did 20 repetitions of the metadynamics. And well, we validated this, I mean, comparing the free energy profiles of the different metadynamics. But our main objective, our main aim here was to see what happens if hypothetically we move the carotenoid through the protein cavity. And this is, I mean, it's only one path possible through the protein cavity. The thing is that we are not considering here the role of the light of the formation of the first photoproduct. So it probably triggers some other changes that are not reflected here in this path, but this path reflects that in different locations of the protein cavity, the absorbance, I mean, the spectral features of the system can evolve, and it is related to the experimental measurements. So I don't know if I responded properly. Just let me know if you have any other questions from here.
Yep. Thank you. There is one question from Professor Joaquín Barroso.
Thank you very much. It was a fantastic talk. Congratulations, wonderful work. It's a fascinating area of research, and I just find it very fascinating that these species can find photoprotections through what is basically a biomolecular machine, right? And in that regard, if I understand correctly, this pigment, this carotenoid molecule, works more as a lock than as an active photoprotective molecule. Am I right?
You mean as the lock in the closed state?
Yeah, as the lock between those two fragments.
Yeah, these two, yeah. In fact, there are some experiments working on with homologs of these two domains separated. In this case, in OCP, they are bind, I mean, by, let me show. Yeah, they are binded by a flexible loop, so they are not completely separated units, but there are some experiments with homologs of each domain, and they can form, they can bind and be together and form what is something like OCP. And it interacts like a lock there inside, holding these two domains together. Yeah, it's really interesting.
But what I mean is that the photochemistry of the carotenoid is of little importance, despite being a photosynthetic pigment. It's of little importance with regard of the photochemistry of the whole unit. Yeah,
totally. And well, in fact, the carotenoid in the whole system, when it is activated in its OCP-R form, when the domains are separate, the carotenoid has a function there when it interacts with the other pigments in the phycobilizome. There is, I mean, the function of quenching this excess energy. Maybe the photochemical function is more important there in that part of the mechanism, when it is going to exert the biological function of quenching the energy. Because it is known that it interacts with other pigments in the phycobilisomes, in the antenna complex.
I see. Thank you very much. Congratulations again.
Thank you.
We have now another question from the YouTube audience, and it reads as following. How do you calculate the minimum free energy path?
Oh, yeah, through the Dijkstra algorithm. I will go back there to that slide. We use the Dijkstra algorithm, and we calculate we put like an initial, we mark this as an initial and a final state of the system. and it calculates the minimum free energy. So it goes to towards the different minimum free energy dots along the path. Yeah, it's going to be tomorrow or after tomorrow in Chem Archive with the details of the paper. Mm-hmm.
Perfect. Here we have another one. So again from YouTube, it reads as, what do you expect for retinal behavior inside this protein? Will be possible translocation and domain separation?
The retinal protein?
Retinal behavior.
I don't know the particularities of that system.
Mm-hmm.
So I'm not familiarized with that system. So I don't know if you want to be more detailed.
I only have the question in the chat, so I don't have more. But yeah, how we can try to give an answer to this one? For example, in this carotenoid, it's possible to have this translocation and domain separation in this case, or it's not?
In the case of retinal,
mm-hmm.
I don't know because I'm not familiarized with the system. Again, I think in the retinal proteins, the carotenoid is, the retinal is covalently bonded to the protein. So probably the process go through a different way. So we should, I mean, take a closer look to that particular system in order to answer that. But I know that I'm pretty sure that it's covalently bonded, the ligand there in that case. So probably in this case, it's not, it only has H-bond interactions, which are of course,
So in the case of the covalent interaction, so in that case, maybe it's not possible to have this translocation.
Yeah, maybe it goes through a different process. Yeah, probably.
Yeah. I hope that we answer the question from that audience. Yeah. So any other question? Yeah, so great, Professor Natalia. Thank you again for your nice talk. It's a very informative topic. I learned a lot of stuff there. So I want to give you thanks in the name of all of our network. And thanks to the audience, for the people here in the Zoom chat, and also via the YouTube transmission. So I hope I extend the invitation for the next session. So I hope all of you will join us in the next webinar of our network. And again, thanks Natalia for your contribution and your support to our network. That the idea is to have a continuous increase in people, try to connect different Latin American researchers in different parts of the world, and at some point we are trying to have a stronger and more connected generation of researchers. Yeah. So if anybody of the audience want to continue asking some things, can contact directly to Professor Natalia. So thanks again. Thanks for the question. It was nice to host this seminar. So see you in the next time.
Thank you very much for the invitation. Let's be in touch.
Of course.
Well,
for your next trip to the south.
Yeah.